Okay, so shall we start? Then review session again. I just wanted to go through some sort of confusing or complicated slides, but you also welcome to ask me question at any time. Um, let's see. So I think the format is the same, right? I mean, we multiple choice and also choose for selection. Also 55 questions. I think the understanding is more important. To try to memorize, right? To try to understand, the material is better. Um, so, so first section that we talk about it. This is biosynthesis of fatty acids and triglyceride synthetic pathway. So lipid metabolism, we learned the synthesis pathway and the breakdown pathway, right? So synthesis pathway like fatty acid synthesis, which usually we term it as the lipogenesis, and triglyceride synthesis. So this lipogenesis and triglyceride synthesis are different, right? So de novo synthesis of fatty acids is a lipogenesis, means that the fatty acids can be synthesized from acetyl-CoA molecules. And triglyceride synthesis is that, the substrate for triglyceride synthesis are the glycerol backbone and 3 fatty acids molecules, right? And then the primary tissues for lipogenesis, are the liver. Liver is a primary tissue, right? And adipose tissue can also synthesize fatty acids, but compared to liver, is relatively, the small in terms of the magnitude or extent. And high-fat diet, we know, is to suppress the decreased lipogenesis, and the mechanism for why high-fat diet decrease lipogenesis. You guys remember the fatty acyl-CoA, long-chain fatty acyl-CoA levels increase, right, and suppresses the key enzyme for lipogenesis. Remember ACC, acetyl-CoA carboxylase. So this is the main reason. And high carbohydrate diet should stimulate lipogenesis. Remember the concept or the definition of lipogenesis is to convert a surplus carbohydrate or amino acids to the fat, right, to the fatty acids. So carbohydrate diet should promote lipogenesis. And what is the purpose why we do the lipogenesis? Just try to convert it, extract carbohydrate or protein amino acids to a fat and store as the triglyceride in the tissue. And in which physiological state does lipogenesis occur? Of course, in the fat condition, particularly high carbohydrate consumption. And the lipogenesis usually takes place in the cytosol, and the building block molecules or starting molecules for lipogenesis or fatty acid synthesis is acetyl-CoA, right? So acetyl-CoA can be used to synthesize fatty acids and also, remember, ketone bodies and cholesterol, right? And it's pretty much similar, the concept that I just described. And this just the overall pictures, how lipogenesis is happening in the body. And basically, you have a high carbohydrate diet, you have an increased glucose level, and that will stimulate insulin secretion and also provide a high substrate levels, which is the glucose, right? So glucose being uptake into the liver and synthesize fatty acids, and glucose can also be taken by the fat tissue and synthesize the fatty acids, but in the skeletal muscle can also have some lipogenesis activities, right? In fatty acids can, lipogenesis can also occur in the skeletal muscle, but this is more for the synthesis of the triglyceride and stored in the skeletal muscle. And the key reaction for the lipogenesis is remember the conversion of acetyl-CoA to malonyl-CoA, right? Malonyl-CoA formation from acetyl-CoA is a key reaction for the lipogenesis, and the two enzyme complexes involved in this, and you just need to remember the key acetyl-CoA carboxylate complex, right, ACC. And also for the fatty acid synthesis, which occur in the cytosol, and the blocking molecules, acetyl-CoA, generated in the mitochondria. Remember, right, from the pyruvate, and so acetyl-CoA need to be transported to the cytosol from mitochondria for the fatty acid synthesis, and this process requires the shuttle system, the citrate shuttle, right? So the purpose of citrate, or the functional purpose of citrate shuttle is to provide acetyl-CoA for for the fatty acid synthesis in the cytosol. So basically, it's transported the citrate from the cytosol, from the mitochondria to the cytosol, right? So you just probably you want to just remember that it's facilitate the acetyl-CoA to move across the inner mitochondria. I think it's the purpose is to move acetyl-CoA from the mitochondria to the cytosol, although it's not directly transport acetyl-CoA, but transport citrate, right? But the purpose is to move acetyl-CoA from mitochondria to the cytosol for lipogenesis. And this is ACC enzyme, key enzyme, catalyzed formation of malonyl-CoA, and this is the two steps, right? The carboxyl group that is transferred to the acetyl-CoA molecule and form the malonyl-CoA, and the carboxyl group initially from the bicarbonate, right? And the two enzyme system that catalyze the transfer of carboxyl group from bicarbonate to acetyl-CoA and for making malonyl-CoA. And also this lipogenesis requires the fatty acid synthesis complex, and you don't need to remember that the whole 7 enzymes, but you know that these 7 enzymes involve the whole fatty acids synthesis process, the adding the 2 carbon to the fatty acyl molecules to extend the carbon chain, right? Carbon hydrogen chain. So this is the whole process, and again, you don't need to remember each detailed reactions, right? But you need to know the whole concept. And the substrate, the acetyl-CoA and malonyl-CoA. Malonyl-CoA is the carbon donor, right? So fatty acid synthesis is going the cycles. Each cycle you add the 2 carbon to the existing fatty acyl molecules to extend the carbon chain, right? And each cycles you use one malonyl-CoA as a carbon donor, right, to add the 2 carbons. And also there is 2 the reduction reactions, right, and that requires the NADH. Each cycle requires 2 NADH, right, to for the reduction reactions. So I forgot the first reaction. First reaction, remember, this is the key reaction, right? ACC catalyzes the formation of malonyl-CoA from acetyl-CoA. So in general, I mean, the fatty acid synthesis required one acetyl-CoA as the first starting molecules, and then for the following cycling, they require the malonyl-CoA as the carbon donor, right? So you probably need to know, to be able to calculate, how many, acetyl-CoA and malonyl-CoA is required to make a one molecules of, like, for example, palmitate or whatever, the fatty acids. Just know that the whole concept, whole process. And the NADH, provision required NADH, right, for the fatty acid synthesis. The main sources of NADH from the pentophosphate pathways, and but it's also the two other sources that can also provide the NADPH and maleic enzyme and isocitrate dehydrogenase. The reactions. You be able to account, for the substrate, how many acetyl-CoA molecules are needed to synthesize palmitate, 1, right, and 7 malonyl-CoA molecules required for 1 molecules of palmitate synthesis, and the 14 NADPH. Yeah.
So we know that palmitate is a 16-protein fatty acid.
Right.
But on the exam, if you wanted us to calculate another fatty acid in terms of the substrate, would you tell us how many carbons were in it?
Yeah. We will tell you, for example, 14, or something like this. Right. Get napkin. So understand this as a whole process is the most important, right? So if you maybe tell you another 16, 18, or 14, you should be able to calculate. And then the regulation, and we talked about it already, right? High fat diet suppress the lipogenesis and insulin, the glucagon and calorie restriction inhibits lipogenesis, and high carbohydrate diet and high sucrose diet and insulin should stimulate lipogenesis, right? And let's just go a little bit detailed the specific regulators of the ACC, which is a key enzyme, right, in the lipogenesis. The allosteric regulation, the citrate, the long chain acyl-CoA. These are two important regulators of lipogenesis. Citrate should stimulate because citrate, that provides acetyl-CoA, right? So if we have a high citrate level, that indicated we probably have a lot of energy, metabolized from glucose or from amino acids. So long chain acyl-CoA is an inhibitor, right? High fat diet, should have high levels of long-chain acyl-CoA that inhibits lipogenesis. And in terms of a hormonal regulation, the insulin basically stimulates, which makes sense. Lipogenesis occurs in the fat condition, right? So insulin should stimulate lipogenesis to store energy, and glucagon should suppress lipogenesis. These two hormones also can regulate the gene expression, right? The insulin, glucose stimulate the ACC expression, and glucagon, cyclo-EMP. Remember, I talk about this. Usually, cyclo-EMP always, connected with the glucagon epinephrine during the fasting condition. So during the fasting, the glucagon epinephrine levels increase that can stimulate the production of cyclo-EMP. So cyclo-EMP level should be high in the fasting condition, and that should promote, the catalytic pathways right? And should inhibit the anabolic pathway, for example, here lipogenesis. So high levels of glucagon or cyclo-AMP should inhibit the lipogenesis, inhibit ACC. Right? And this just give you a little bit the more information about AMPK. So AMPK, you can also link AMPK to cyclo-AMP or glucagon and epinephrine. During the fasting, the AMPK level or AMPK activity, AMPK is a kinase enzyme, right? So AMPK activity should be increased. So AMPK can phosphorylate ACC. So just one more level regulate ACC. So AMPK can phosphorylate ACC and decrease ACC activity, right? So when AMPK is activated, it should inhibit lipogenesis, right? Inhibits ACC activity. So again, connect AMPK to the fasting condition, epinephrine, glucagon. And triglyceride synthesis and basically is synthesized triglyceride molecules from 3 fatty acids and 1 glycerol phosphate, and glycerol and 3 phosphate as the substrate, right, for the triglyceride synthesis. And this and triglyceride synthesized first, fatty acids should be activated to fatty acyl-CoA, and this reaction is actually the common to the fatty acids oxidation, right? So both fatty acid oxidation and fatty acid synthesis, the first step, fatty acids should be activated to fatty acyl-CoA. So this is a common reaction to both fatty acid synthesis and fatty acid oxidation, right? And this is the enzyme fatty acyl-CoA synthetase. And the sources of a glycerol 3-phosphate for triglyceride synthesis mainly from glycolysis, right? And the other sources is of glycerol 3-phosphate can be directly generated from the glycerol phosphorylation and by glycerol kinase, and this only occur in the liver because glycerol kinase only present in the liver, not in adipose tissue, right? So in the liver, the glycerol 3-phosphate should have came from the 2 sources, right? One is from glycolysis and the other from directly phosphorylation by glycerol kinase. And in the adipose tissue, the glycolysis is the only way to generate the glycerol 3-phosphate. And the transporter. So once the triglyceride synthesized in a different place, for example, and triglyceride can also be synthesized in the small intestine, right? Once we absorb the free fatty acids from the diet, and free fatty acids be resynthesized to triglyceride, and triglyceride need to be transported to the adipose tissue for storage. So the small intestine, we also learned the lipoprotein, right? So lipoprotein in small intestine, which is a major lipoprotein, chylomicron, right? So chylomicron is a main lipoprotein that's synthesized in the small intestine, can pack the triglyceride and then transport the triglyceride from small intestine to the adipose tissue for storage. And the liver, the triglyceride can also be synthesized from the lipogenesis pathway, right, in the fat condition, and packed into the VLDL and transported into the adipose tissue for storage. This 2 transported pathways usually occur in the fat condition. And during the fasting, triglyceride can also be packed into the VLDL in the liver, but it's usually transported to the muscle, and muscle pick up the fatty acids from the VLDL triglyceride and utilize as energy, right? So again, different physiological condition can have a different route of the transport of triglyceride, and have a different metabolism, metabolic fate. And we also talk about the LPL, right? It's very important, the lipase, and LPL basically hydrolyze triglyceride in both chylomicron and the VLDL, and release fatty acids, and fatty acids then be picked up by the adipose tissue for resynthesized triglyceride and store as energy in the fat condition, right, again. But in the fasting condition, muscle VLDL can hydrolyze triglyceride in VLDL and pick up fatty acids, and fatty acids being oxidized for energy, right, in the fasting condition. So again, we know that LPL can be produced from the two different tissues, adipose tissue and muscle, right? And the synthesis or secretion of LPL and can be regulated differently in different tissue, right? Fat tissue, insulin, or fat in the fat tissue, insulin is the is an important hormone that stimulates LPL synthesis and secretion from fat tissue in the fat condition, right? And muscle is synthesize or secrete fatty LPL by the during the fasting condition, right? So fasting condition LPL in the muscle can be synthesized more and secreted more. It depend on the tissue and physiologic condition. LPL can be regulated differently, right? All right. Yeah, in addition to fasting, exercise can also stimulate LPL, muscle LPL secretion, right? Synthesis. Okay. Now let's talk about the oxidative pathway, fatty acid oxidation. So, and ketogenesis. Fatty acid oxidation, ketogenesis usually occur in the fasting conditions, right? So before we talk about the oxidative pathway, I just wanted to go through again the storage of triglyceride in the fat condition, right? So dietary fatty acids, when we consume a lot of diet, and dietary fatty acids can be absorbed by the small intestine and re-acidified to triglyceride in the enterocytes, and triglyceride being packed into the chylomicron and transported to the adipose tissue for storage. And again, the LPL is very important. Lipase hydrolyzed triglyceride in chylomicron, release fatty acids, right, and provide fatty acids for triglyceride synthesis in the adipose tissue. And liver also can synthesize a lot of fatty acids from the carbohydrate glucose during fat condition, right, through the lipogenesis pathway. And fatty acids, once it's synthesized, it's also re-acidified to triglyceride and packed into VLDL. And that, again, the protein lipase is secreted from fat tissue. It's also important, hydrolyzed triglyceride in VLDL once VLDL arrive in the adipose tissue, right? And then fatty acids is also be picked up by the adipocyte. So the fatty acids for triglyceride synthesis in the adipose tissue primarily from the diet, right, from the small intestine and absorption, and also from the lipogenesis in the liver. And then the 3-glycerol 3-phosphate, the backbone, basically is from the glycolysis pathways, right? In the adipose tissue, see the dietary carbohydrate can be uptake by adipose tissue. This is a very important transporter, group 4, depend on insulin, right? So uptake group 4 increased by insulin stimulation in the fat condition. So glucose can be metabolized through the glycolytic pathways and provide the glycerol 3-phosphate. And of course, some of the acetyl-CoA that is metabolized from glucose can also be used for fatty acid synthesis, right, through the lipogenesis pathway. But again, this contribution relatively less compared to the fatty acids that uptake from the circulation, right, initially from the small intestine and the liver, right? Okay, so now mobilization. So when we during the fasting condition, when we don't have enough food available or enough nutrient, particularly fatty carbohydrate or fatty acids, then we need to mobilize the the stored energy. And since we talk about lipid metabolism, right? Actually, glycogen is also stored, right? So we mobilize glycogen. But for the fat, we mobilize the fatty acids from adipose tissue. So we break down the triglyceride to fatty acids and release fatty acids. And fatty acids can be picked up by multiple tissue and utilized as an alternative energy fuel, right? And also because the brain cannot pick up fatty acids and utilize other energy, so our body, the liver, can make ketone bodies, which much smaller molecules. So ketone body can be picked up by the brain, but just by simple diffusion mechanism. And so brain can utilize the ketone bodies as an alternative fuel instead of fatty acids, right? But muscle be able to utilize both fatty acids and the ketone bodies for energy. So during the fasting conditions, we need to break down triglyceride, which the process we call the lipolysis, right? Remember, and to release fatty acids, and we also need these to synthesize ketone bodies. And again, the ketone body, the substrate for ketone body synthesis is the acetyl-CoA, right? So fatty acids is first of all picked up by the liver and break down through the sort of called a beta oxidation, right, to make a acetyl-CoA, and then acetyl-CoA can serve as a substrate for ketone body synthesis. So here is the mobilization of triglyceride, or we call it lipolysis process, right? Basically, it's break down triglyceride to release fatty acids. And the key enzyme for lipolysis is, you guys remember the key enzyme? HSL, hormone-sensitive lipase, right? Although there are other lipase involved, but I just want you guys to remember the HSL is the key enzyme to break down the triglyceride into the fatty acids, right, and glycerol. So fatty acids then be released. And the regulation of this key enzyme HSL, basically the two hormones, two sets of hormones, right? One set of hormone usually is increased during the fasting, which are epinephrine and glucagon. And as I mentioned earlier, that glucagon epinephrine always connect to the cyclo-AMP, right? Can increase the cyclo-AMP and stimulate protein kinase A. And protein kinase can phosphorylate HSL and stimulates HSL activity, right? So and then can break down the triglyceride. And the other set of enzyme is the hormone C, insulin. So insulin during the fasting condition is actually decreased. So insulin decrease can dephosphorylate HSL and leads to inactive, the inactivate HSL, right? So these two sets of hormone work together to make the HSL activated, right? All right. So okay, so the lipolysis, in addition to HSL, ATGL and TGL can also involve in the whole, the complete breakdown of triglyceride to release 3 fatty acid molecules require this three lipase, right? But again, we just wanted to remember this HSL is a key enzyme. And once fatty acids is a breakdown and released from adipose tissue and picked up by the tissue that utilized for energy, for example, muscle and liver, then fatty acids undergo oxidation, right, to further break down long chain, fatty acids to smallest molecules through the beta oxidation. So which is the smallest molecules that after fatty acid oxidation, beta oxidation? Do you guys remember? Yeah, acetyl-CoA. Remember, we were going to test them Thursday, right? So acetyl-CoA is the last and the smallest molecule, right, after beta oxidation. So if in the muscle, then acetyl-CoA molecules can be further oxidized to CO2 water and to produce ATPs, right, through enter the TCA cycles, remember, right, can be further oxidized. But in the liver, acetyl-CoA that generated from beta oxidation of fatty acids will use for majority of the fatty acetyl-CoA, right, generated from beta oxidation in the liver are used for synthesis of ketone bodies, right? So again, this is the first reaction, which is the same as the triglyceride synthesis, right? Fatty acids, first of all, need to be activated to fatty acyl-CoA, which occur in the cytosol, and through this, by this the enzyme, fatty acyl-CoA synthetase, right? Again, this reaction occurs in the cytosol. And then the next step is the beta oxidation. So beta oxidation, remember, occurs in the mitochondria, right? So fatty acids, once is the activated to fatty acyl-CoA and need to be transported into the mitochondria for beta oxidation because the sets of a beta oxidation enzyme is in the mitochondria. So the transporter, again, this beta oxidation process, the final product of beta oxidation is acetyl-CoA, right? So palmitate has 16 carbons and break down, oxidize through beta oxidation, generate 8 acetyl-CoA molecules, right? So again, this beta oxidation also goes cycles. Each cycle you remove the 2 carbon, right, as the acetyl-CoA molecules. And transporter of fatty acyl-CoA into the mitochondria, and CPT1 again is a key enzyme to catalyze this process. So transport of fatty acids controlled by CPT1. So and I don't think you need to remember the whole detail, but again, try to remember this key enzyme. What is the key enzyme does is to transport the fatty acyl-CoA from cytosol into the mitochondria for beta oxidation, right? So beta oxidation can be controlled by the CPT1 activity, right? If you have a higher CPT1 activity, you can transport more fatty acids into mitochondria, so you have a more substrate for beta oxidation, right? So increase. So this again, the CPT1 activities regulates the beta oxidation rate. And I think this diagram is very important, very helpful for you to understand the whole process. How fatty acids is oxidized to produce the ATPs and also CO2 water. And this diagram represents the complete oxidation fatty acids, right? So start with activation, fatty acids or fatty acyl-CoA, and then transport fatty acyl-CoA into the mitochondria controlled by CPT1, right, mitochondrial membrane. And then the beta oxidation process eventually generate the final product, acetyl-CoA, and acetyl-CoA further oxidized enter the TCA cycle for complete oxidation, right? And so you try to remember this whole process and be able to calculate the NADH or FADH or ATP production. Remember the beta oxidation process also generated one FAD and one NADH molecule, right? So and it's just each cycle. One cycle you generate one FAD and one NADH, and if complete oxidized palmitate, which is 16 carbon molecules, you will go how many cycles? Remember, 7 cycles, right? And generate 8 acetyl-CoA molecules. And one acetyl-CoA molecule enter TC cycle generate how many ATPs? 12, right? And then you will have 8 acetyl-CoA molecule enter TC cycle, right? From the oxidation of palmitate. So try to remember the whole process. So I think this will help you to understand the whole process by calculating the ATP production, right? Yeah, it's just calculation of the ATPs. Now ketone body synthesis. So ketone body synthesis when occurs in which physiological condition? Fasting, right? Usually it's a prolonged fasting, like fasting for longer than 2 days, then our body starts to synthesize a significant amount of a ketone body. So in other words, in the fat condition, we don't have high levels of ketone bodies, right? It's not detectable ketone body. But when fasting, prolonged fasting, then ketone body levels increase. And the place that, or the tissue that synthesizes ketone body is the liver, right? So liver is the only tissue that can synthesize the ketone bodies. And also I wanted to mention, in addition to the prolonged fasting, the fatty acids level increased, for example, during in the diabetic conditions, the high levels of fatty acids in the circulation under this condition, ketogenesis also increased, right? So under the pathological condition, like diabetic condition or alcoholism, ketogenesis will also increase. So liver is the is the organ tissue that synthesizes ketone bodies, and liver usually is not able to utilize the ketone bodies. The reason you guys know, right? The lack of enzyme. Remember that enzyme? Succinyl-CoA transferase, right? Okay. So and ketogenesis again it usually is started with the acetyl-CoA molecules, right? And the first reaction that catalyzed the catalyzed by HMG-CoA to generate HMG-CoA by HMG synthesis, and this reaction is the same reaction as the, remember, the cholesterol synthesis, right? So cholesterol is also synthesized from acetyl-CoA. So the first two reactions, I think, for the synthesis of HMG-CoA is the same for both the ketogenesis and cholesterol synthesis. Here just the synthesis of the ketone bodies from the acetyl-CoA, which initially from the fatty acids oxidation during the fast condition, right? We know that acetyl-CoA can also be generated from glucose metabolism, but because under the fasting condition, we don't have much glucose, right, to be break down to the acetyl-CoA because we try to spare glucose. So usually glucose just goes glycolytic pathway and stop at the pyruvate, right? Or not for the acetyl-CoA production. So acetyl-CoA that utilized for ketone bodies primarily from fatty acid oxidation, right, during the fasting condition, but also during the diabetic or alcoholism, and synthesized ketone bodies being released into the circulation and picked up by tissues, muscle, brain, and some other tissues. And utilization of ketone bodies, once it's uptake by tissue, here we just use the muscle as an example, and ketone body can be converted back to the acetyl-CoA, and this is a key enzyme, the acetyl-CoA transferase, right? And this reaction requires 1 ATP for as energy for the ketone body utilization. So and liver does not have this acetyl-CoA transferase, so cannot utilize ketone bodies. In terms of regulation of ketogenesis, I think the three key points that can regulate the ketogenesis are very important. First of all, the lipolysis and fatty acids levels. So, for example, during fasting condition, lipolysis increased to break down triglyceride and release a lot of fatty acids. So fatty acid level in the circulation is increased. So this increase can promote lipogenesis. Promote ketogenesis. And the second level is the CPT1, right? CPT1 is a key enzyme control transport fatty acids into mitochondria for beta oxidation. So can control beta oxidation. And last one is the acetyl-CoA levels. So beta oxidation we know is can generate acetyl-CoA, right? So if beta oxidation is increased and acetyl-CoA levels increase, then can provide a more substrate for ketogenesis. So again, the fatty acid level in the circulation, CPT1 activities on the mitochondrial membrane, and beta oxidation generated acetyl-CoA levels. Three things can control the ketogenesis, right? So try to remember it's the control, the regulatory mechanism point, and this just the two conditions, the ketoacidosis. One is the diabetic condition, right, and the other is the alcoholism, and because of these two conditions have some problems with the glucose utilization, particularly in the diabetic condition. We know that the transport glucose into the tissues is a problem, right? Insulin resistance. So glucose cannot be taken up by the tissue and utilize energy. So theoretically, the cell is under the energy crisis. So under this condition, that body need to promote more fatty acids and break down and utilize as an alternative energy. So fatty acids levels increase in the circulation in both diabetic and alcoholic subjects. So then we know that fatty acids increase is one of the important drivers, right? Remember, for the ketogenesis. So you have a more acetyl-CoA produced, and then promote ketone body synthesis. And these are the two very important slide, I think, in terms of the regulation of the fatty acid oxidation. Both actually fatty acid oxidation and the lipogenesis, right? Let me see. Let me finish this two slide, and then we take a break, right? So in the fat condition, what do we expect lipogenesis? Should be increased, right? And how about fatty acid oxidation or lipolysis? It should be decreased, right? We don't want it to break down fatty acids, and we have enough, energy substrate from the diet. So we need to stimulate lipogenesis to store excess energy that we get from diet, and we should shut down the lipogenesis of fatty acids breakdown pathways. So in the fat condition, the glucose levels increase, right? And acetyl-CoA be generated from glucose oxidation, and this is the lipogenesis pathway, right? Acetyl-CoA converted to malonyl-CoA and ACC. So for this regulatory mechanism, and try to focusing on the ACC that control lipogenesis, and the malonyl-CoA and CPT1 that control the fatty acid oxidation, right? So the ACC in the fat condition, insulin levels increase, so insulin stimulate ACC, right? And so then we have a more malonyl-CoA produced in the fat condition, because malonyl-CoA is the inhibitors of CPT1, right? Remember this. Malonyl-CoA is inhibitors of CPT1. So increased malonyl-CoA level in the fat condition will lead to decrease the CPT1 activities, right? Then you have a decrease the transport of fatty acids into the mitochondria for oxidation. So then beta oxidation will be reduced if CPT1 decrease, right? So this is something happening in the fat condition. And in the fasting, we don't want to synthesize lipids, right? Fatty acid. We wanted to utilize fatty acids. So we expect that the lipogenesis would be decreased, but lipolysis of fatty acid oxidation pathway will be increased. So in the fasting conditions, because of lipolysis is increased, right? So we have more fatty acids released from adipose tissue into circulation. So increased fatty acids level increased fatty acids level in the fasting condition will causing increased fatty acyl-CoA levels, and fatty acyl-CoA is the inhibitors of ACC, remember, right? So then ACC activity will be decreased, and lipogenesis will be decreased. So because of ACC decrease, the malonyl-CoA levels decrease. So this inhibition will be will be released or will be gone, right? The inhibition of the CPT by melanocortin will be gone because melanocortin levels decrease. So that will lead to increase CPT1 and then increase the fatty acid transporter and beta oxidation and increase the ketogenesis, right? So try to again remember these two slides and focusing on the the two lipogenesis and lipolysis fatty acid oxidation, these two opposite metabolic pathways, and the two key, actually three key things. The ACC, the key enzyme for lipogenesis, and CPT1 is the key enzyme controlling fatty acid transport and oxidation, and the melanocortin, which is the regulators of a CPT1 activities, right? So, and see how, in fasting conditions, how these key things change and then regulate the two different pathway activities, right? Okay, so let's take maybe 10 minutes break. Okay. Do you guys want me to, did I talk too detailed too much or something? You don't want me to? I don't know. I have a lot of slides actually. I wanted to have some time for you guys to have questions, so and just try to go through a little more, focus on kind of more major point, right? Not go to too details. So okay, so now the cholesterol synthesis. I just talk about the common pathways, right? The first, reaction, they are the same as the ketone body synthesis. Both ketone body synthesis and cholesterol, they use the same acetyl-CoA, right? The as a molecule. So, and HMG-CoA reductase is a key enzyme for cholesterol synthesis, and just try to remember this, and which are the inhibitors and stimulators, of the cholesterol HMG-CoA synthetase. And I think we have talked about the substrate, right? And I think the drug that the statin is the inhibitor of HMG-CoA to reduce the synthesis of cholesterol. And the 17 enzyme, that's just a whole process. You don't need to remember all the enzyme, but just substrate, acetyl-CoA. Again, the HMG-CoA reductase, a key enzyme, and regulation. Probably want to just focusing on this two substrate, the cholesterol and malonate, sorry, the inhibitor, right? The HMG-CoA reductase. The cholesterol synthesis? No, just the key enzyme HMG-CoA reductase. And that's a regulated mechanism, just in adding more levels of transcription factor. SIEBP is the target of the cholesterol and malonate, right? Malonate inhibits the SERBP transcription factor activity, so then reduce the gene expression HMG-CoA reductase. And the lipoprotein lipase, lipoproteins, right? Lipoproteins, and the structure that the core is the lipids, basically is the esterified lipids, right? Triglyceride and esterified cholesterol, and the surface has the phospholipids, monolayer phospholipids, and free cholesterol, and the apoproteins. So apoproteins, we have there's a variety of different apoproteins, and the function. And I think you wanted to focus on this. And some apoproteins can have a function as a co-activators of enzyme. For example, ApoC2. Right? ApoC2 is a co-activator of a lipoprotein lipase, LPL, and also the ApoE and ApoB100 are the important regulators of the LDL receptor binding, lipoprotein binding to LDL receptors, right? So try to remember this two sets of apoprotein, what are their functions, right, in terms of the regulation of lipoprotein metabolism, and also some kind of unique lipoproteins. For example, ApoB40 is unique to chylomicron and synthesized in the small intestine, and B100 is unique to VLDL, right, and synthesized in the liver. And also the two different type in terms of lipids content. One type of lipoprotein called triglyceride-rich, right, lipoprotein, like chylomicron and VLDL, and LDL, HDL is cholesterol-rich. So that means it's the chylomicron, VLDL basically regulate or transport a triglyceride, and you need to remember each chylomicron, where they synthesized and what is the function, how they regulate triglyceride metabolism, transporter, or cholesterol metabolism, right? So LDL and HDL usually is the cholesterol rich, the carmicron means that they regulate cholesterol metabolism, basically. And each important, the apoprotein on the each lipoproteins, right? Try to remember, like ApoB40 and on the carmicron, ApoB100 on the VLDL, and ApoC2 on both carmicron VLDL, which makes sense, right? Because it's regulate like LPL, and LPL hydrolyze the triglyceride metabolism. So ApoC2 should be on the triglyceride-rich carmicron and VLDL and regulate triglyceride levels, right? And ApoE and B100 present almost on the all different type of the lipoprotein except for the HDL. And remnant lipoproteins, the carmicron remnant usually pick up by the liver, right? And the triglyceride in the carmicron remnant hydrolyzed by liver, like hepatic lipase. And VLDL remnant are the IDL or LDL, right? And pick up by other tissues. And this I just talked about, you go through the all different individual lipoproteins and where they're synthesized and what is the function, and what kind of apoproteins on their surface and what the specific apoprotein does, function or something like this, all right? Okay, let's see. And the metabolic fate of each individual lipoproteins, and just quickly go through this. Like carmicron basically synthesized in the small intestine, right, and pack the triglyceride from the diet and transport the triglyceride from intestine to the adipose tissue. And again, the LPL is important to hydrolyze triglyceride in the carmicron and carmicron remnant and pick up by the liver. And VLDL is another, the triglyceride-rich lipoproteins, and the synthesized in the liver and pick up by the adipose tissue, and same thing, and regulated by LPL and ApoC2, and is also very important for LPL activities. And you also need to know that once the ApoC2 deficiency will causing the problems, right? We mentioned this in the class that we increase, for example, ApoC2. A deficient patient will have a higher levels of triglyceride, right? And also high levels of carmicron and VLDL. And the LPL, in terms of function, is to hydrolyze triglyceride in both carmicron and VLDL. And you probably wanted to compare the LPL in muscle and adipose tissue, and in terms of the regulation, the difference between these two tissue LPL. Yes. Oh. Me here? Back more or the? Oh, here. Oh, okay. Oh, you mean this here? Oh, how triglyceride in VLD and carmicron metabolized? Okay, so triglyceride, both actually hydrolyzed by LPL, right? So then release fatty acids, and fatty acids then be picked up by adipose tissue. So this is sort of kind of process how triglyceride be break down by LPL in both VLDL and chylomicron. And second question, which enzyme is the key for LDL chylomicron to chylomicron triglyceride metabolism? Is LPL, right? LPL. Okay. Yeah. Oh, ApoC. ApoC, because ApoC2 is required for LPL activity, right? So ApoC2 deficiency patient then you have a lower LPL activity. So that means they are not able to hydrolyze triglyceride in VLDL and chylomicron, right? So a lot of triglyceride remain in the circulation, cannot be, taken by the adipose tissue. So causing increased triglyceride levels and also increased VLDL and chylomicron, right? Right. Okay, so yeah, this just basically is the LPL. Depend on the fat or fasting condition. So in the fat condition, the LPL is secretion increase from adipose tissue, right? And this LPL usually hydrolyze triglyceride in both chylomicron and the VLDL and release fatty acids. And in the fasted condition, because catecholamine like epinephrine or glucagon increase, so that can promote lipolysis, fatty acids or triglyceride mobilization. So triglyceride breakdown by HSL, right, hormone-sensitive lipase, and then release fatty acids. And fatty acids can be picked up by the liver to synthesize triglyceride. And this synthesized triglyceride can be packed into VLDL and then picked up by muscle. Right? During the fasting, muscle LPL activity is increased. But liver, the adipose tissue activity, LPL activity is not active, right? So the LPL in the muscle can hydrolyze triglyceride in VLDL and then pick up fatty acids. And this fatty acid, once it pick up, usually used for oxidation for energy, right? So it's quite a different from the adipose tissue in the fat condition. So fatty acids pick up by adipose tissue in fat condition through the LPL hydrolysis of the triglyceride is to re-acidify triglyceride again, right? Stored in the adipose tissue. So and then the two other important lipoprotein, LDL and HDL. What you need to know is, I think it's important that the ApoB and ApoE, which are on the LDL surface and very important to for the LDL pickup by the LDL receptor, right, into the tissue, into the liver or the peripheral tissue. So liver is the most important tissue that pick up LDL. So if something is wrong with the ApoB100 or ApoE, then the LDL cannot be recognized by LDL receptor, right? So then LDL cannot be picked up by the tissue, liver or peripheral tissue. Then the LDL level will be increased. So, and we also connect this to the atherosclerosis, remember, right? So again, the LDL receptor number and also this ApoB100 100 ApoE protein function is very important, regulate LDL uptake and LDL levels in the circulation, right? For some reason, if LDL receptor number somehow is relatively low, so some genetic reason, or the ApoB100 ApoE has some problem, then LDL cannot be picked up efficiently by the liver primarily, right? So then can raise the LDL level in the circulation. It's not the cholesterol level itself, it's actually the LDL number, receptor number, and also the ApoB100 ApoE protein, apoprotein function is most important that control or regulate the plasma LDL cholesterol levels, right? So, and it's just a receptor, the mechanism usually, LDL, the whole lipoprotein bind to LDL receptor and internalize into cells. And we also talk about the HDL cycles. HDL is very important, right? And the main function of HDL is to do the reverse cholesterol transport, meaning that the transport the unnecessary free cholesterol from peripheral tissue back to the liver for the excretion, right? So to the cholesterol level, we need to maintain the balance in normal levels. We don't want too much cholesterol. We need cholesterol, but we don't want to have too much. So the HDL cycle is served as a function to dispose the extra cholesterol from the peripheral tissue, so then can prevent the cholesterol deposition in the peripheral tissue if it deposits in the macrophages, right, when causing the problem, the firm cell formation, atheroma formation, and the atherosclerosis that can occur in the artery wall. So HDL, and you wanted to know that the conversion between beta HDL and alpha HDL, right, to accomplish the HDL cycles. So pre-beta HDL basically is is it called empty? Remember what is it called? Empty dump truck? No, not dump truck. It's truck can load it. Can load the cholesterol, right, from the peripheral tissue. So in the peripheral tissue, the beta HDL have higher levels, higher concentration. So the free cholesterol can be transported from peripheral tissue to the beta HDL. So beta HDL usually don't have much lipids, right? Like empty, so the beta HDL once have a free cholesterol or phospholipids from the peripheral cells and then converted to alpha HDL. It is alpha HDL that transport cholesterol to the liver. So and also there is the this is a slide that I just described, right? And the alpha HDL and be picked up by the liver, and also the several the process that occur in the plasma and during the conversion from beta HDL to alpha HDL is very important. Remember the enzyme that you guys need to remember. Couple enzymes. For example, here the ABC1 is the transporters, right, of free cholesterol. So can transport free cholesterol from peripheral cells to the beta HDL. And again, you also need to know the genetic problem with the ABC1 deficient or some male dysfunction. Question. I think so. I did post it, but I can post actually this review, slide, right? So the ABCA1 deficiency, it will also causing some problem, right? Because we cannot transport free fatty acids to the beta HDL. So you will have a lot of free fatty acids still remain in the peripheral tissue, cannot be, transport out. And then the pre-HDL will have a high levels because we have a no free cholesterol being loaded, right? And then have a lower levels of alpha HDL. Beta HDL cannot be converted to alpha HDL without free cholesterol being loaded from peripheral cells to the alpha HDL. And also there is the reacidification. Once the free cholesterol pick up from free cell peripheral cells and need to be reacidified to cholesterol and then can be internalized to the core of the beta HDL, right? And so the enzyme, the LCAT, is the enzyme that catalyzes reacidification of free cholesterol to the cholesterol ester in the HDL, right? In this reaction occurs in the plasma, and there are also another enzyme called ACAT, right? ACAT has the same function as the LCAT, but the location is different. ACAT is located inside of cells, right? And then the exchange CEPT is a protein enzyme that can catalyze the lipid exchange between HDL and VLDL, IDL, and LDL, right? So try to remember these two different protein enzymes, how they, what kind of function that they have, or something like this, right? And this is similar. This I just talk about, and this just showing the lipid exchange. So basically, triglyceride being transferred from VLDL, LDL, IDL to HDL, and cholesterol esters and transferred from HDL to this lipoproteins, right? So about 30% of cholesterol esters in the VLDL actually be transferred to the VLDL, IDL, and LDL, right? And once it's cholesterol transported from peripheral tissue to the liver, and it can be used for synthesis of bile acid and excreted, right? And also can be used for synthesis of steroid hormones. And here is the slide. I just talk about the ApoC2 deficiency, right? Can cause an increase the cholesterol. Increase the triglyceride, chylomicron, and VLDL levels in the circulation, right? The reason, right? Okay, now fatty liver disease, and just because of the imbalance between, synthesis and the export of triglyceride, so causing the accumulation of triglyceride in the liver. And a couple of reasons that first of all, synthesis of triglycerides increase. For example, the diabetic conditions and chronic intake of the high-fat diet, you have a lot of fatty acids in the circulation available and picked up by the liver and synthesize triglyceride. So this is one of the reasons that can cause fatty liver disease. And the other reason is that once the triglycerides are synthesized in the liver, if they cannot be transported out of the liver, then will also cause an accumulation, right, or build up in the liver. So anything that affect the lipoprotein function, that will causing the problem that triglyceride cannot be export. For example, somehow the blocks in the apoprotein synthesis. Some of the important apoprotein that cannot be synthesized, so cannot be make functional lipoproteins, and also the lipids, the phospholipids, if for some reason cannot be provided, it's also causing the problem because the surface of lipoprotein requires the phospholipids, right? And it impairs the secretion of lipoprotein. For some unknown reason that the synthesized lipoprotein cannot be secreted, right? So all these problems can affect the export of triglyceride. And homeostasis, the cholesterol levels in the plasma, I just talked about it. The LDL receptor numbers, or the ApoB100 and ApoE functions, very important, that regulates uptake of LDL. So decreased LDL receptor or defective ApoE or ApoB100 can causing the decreased binding of LDL to LDL receptor. So then have a lot of LDL cannot be picked up or remain in the circulation and raising circulating levels of LDL. So again, the LDL receptor is the main regulating mechanism, affecting the LDL level in the circulation, right? And inside of cells, also there is balance, right? Cholesterol can be maintained at the normal range inside of cells, and there are several pathways, can regulate the levels of a free cholesterol pool inside of cells, and synthesize, uptake, and the acidification process that can also contribute to the levels of free cholesterol. So I listed here that 5 different reasons that can cause an increased cholesterol level inside of cells, right? For example, if uptake too much LDL cholesterol from the circulation, or synthesize more inside of cells, or increase the hydrolysis of cholesterol esters, or decrease the export or efflux, and which this is very important, right? The ABCA1 transporter is a very important protein that regulates the export of free cholesterol, or the synthesis. If somehow cholesterol cannot be utilized to synthesize some sterols or some other molecules, then this will all affect the cholesterol homeostasis or levels inside of cells. So try to remember this, the different pathways, how they affect the cholesterol level inside of cells. And atherosclerosis, and I talked about it a little bit already, and the major reason of risk factor for the development of atherosclerosis is the elevated plasma cholesterol level, right? And not related to triglyceride level. So cholesterol level increase is a major contributor to the atherosclerosis, right? And also we talk about like several other reasons. I just wanted to go to this slide. First of all, is the high levels LDL. If for some kind of genetic reasons we already mentioned, right, like ApoE deficiency or ApoB100 deficiency, then you will have a higher LDL level in the circulation. So this is one important reason, or the causes, right, for the atherosclerosis. And the other 2 important causes of atherosclerosis is the artery wall damage, maybe due to the chronic, high blood pressure. So an artery wall damage and attract the macrophage and other immunocells to the damaged site and causing the inflammation. So artery wall damage, inflammation, plus the high levels of LDL, and are the most important causes the atherosclerosis. Yeah. Okay. This one? Oh, okay. Uh, yeah, I mean, this I mentioned, right? The high levels of cholesterol or LDL cholesterol is a major driver, right, or major contributor to the atherosclerosis, and not the triglyceride level, right? Triglyceride level is not the contributors. Um, and then atheroma formation in the, in the intimate arteries is because of the deposition of the cholesterol esters, and this is the cholesterol deposition in the macrophage cells, right, and form the firm cells. So you have a lot of firm cells that filled with the cholesterol that eventually can form the atheroma. So atheroma then can block the artery, block the blood flow of the vessel, right? So and eventually, causing the problem. So I think this slide probably will be best slide for so covering all the process. And these are the three reasons, right? Three the causes that I just mentioned. And so, for example, first of all, you have high levels of LDL, and which can cause by some genetic problems like ApoE deficiency or ApoB100 deficiency, and then plus the damage of an artery wall and macrophage and monocyte infiltration, and then the increased LDL in this damaged site can be oxidized to oxidize LDL. And oxidized LDL can be picked up by a scavenging receptor, remember, right? So then you have a lot of cholesterol being picked up by the macrophages and accumulate in macrophages and form the firm cells. So I have a lot of firm cells that, and together form the atheroma. And also, I think we mentioned the three genetic disease, right? First of all is the tangent disease, and basically it's due to the mutation in the ABCA1 gene. ABCA1 we know that is important, right, for the transporter of the free cholesterol to pre-beta HDL. So, and also we have a couple questions asking whether the alpha HDL level, it should be decreased, right? Because we don't have, the free cholesterol being transported to beta HDL. So beta HDL cannot be converted alpha HDL. So have a lower or absent alpha HDL in the plasma, but have very high levels of beta HDL, right? And risk for the atherosclerosis is high because of the, you have you're not able to remove the cholesterol from the peripheral cells, right? Because of lack of ABCA1, so you have a lot of cholesterol you're not able to remove and accumulate in the peripheral tissue, particularly for example in the macrophages, and can form the firm cells and causing atherosclerosis, right? That makes sense. And then the other two, and the second disease that genetic disease that we talk about is the ApoB100 deficiency, right? This problem that the LDL lipoprotein cannot be recognized by the LDL receptor. So LDL receptor cannot pick up enough LDL from the circulation into the liver, then this causing the increased LDL levels, right, in the circulation, and also increase the VLDL remnant, because LDL is converted from VLDL, right? It's a remnant of VLDL. So VLDL cannot be removed, and then the IDL level will also be increased. And so the cholesterol, LDL cholesterol levels is increased, and risk for the atherosclerosis because LDL levels increase. This is one of the important causes of atherosclerosis, right? So the oxidized LDL can be picked up by scavenger receptor, which is independent of ApoB100. Even the LDL do not have a ApoB100 in this patient, and this LDL still be able to pick up by scavenger receptor, but not be able to pick up by LDL receptor, right? So that's this. You have a more, LDL being picked up by the macrophages, causing problem. And similar to the ApoE deficiency. So ApoE deficiency that remember the chylomicron remnant and the VLDL remnant contain the ApoE. So ApoE deficiency that chylomicron and the VLDL remnant will not be able to pick up. So you have the patient ApoE deficient is a little bit different from the ApoB100 deficient patient, right? So and in ApoE deficient patient, their chylomicron and the VLDL remnant levels in the circulation will be high, right? Because this uptake of remnant, chylomicron, and VLDL requires ApoE, right? ApoE deficiency and causing the accumulation of this remnant in the circulation. And also you have IDL and VLDL levels, right? The VLDL remnant, the IDL and LDL are the VLDL remnant, right? So cholesterol level in the plasma is increased, and the similar reasons, and you also have a high risk for the atherosclerosis. I guess that's all slide I have, and now you guys have any questions? Yeah. Oh yes. Essential fatty acid, yes, will be, because I saw the essential fatty acid is relatively simple, so I did not include it here. Yes, you try to remember the two essential fatty acids, right? Linoleic acid, alpha-linoleic acid. So, and the reason why we cannot synthesize because we lack of delta 6, 12 and 15 desaturates, right? And delta 5 and 6 desaturates we have, so in the body, so we be able to synthesize, omega-3, other family of omega-3 and omega-6 fatty acids. Yes. Sorry, the quiz. Oh, practice. No, we don't have a practice for the lipid metabolism. And the last one I just sort of, let you guys get a sense of the format of the exam. So we don't have a specific. Yeah.
Are you going to post the last quiz that we took, quiz 4?
Quiz 4? Yes. Oh, she has a quiz 4. Yeah, you can come here. So, yeah, the key. Okay. I think probably today we'll post it. Yeah. Any other questions? No? Okay, so. But just feel free if you have a question during study and you wanted to ask me, you can, you can still have today and tomorrow, right?